Okay, today I'm going to be talking about plant diseases and other plant disease happenings here in South Dakota. We're going to be focusing on corn diseases and soybean diseases that my lab group has been tracking and developing diagnostic tools for over the last few years. Tar spot, as most of us have heard of, at least in some capacity, much like in other parts of the country, is becoming a fairly major issue here in South Dakota. In 2022, we first found it in 6 counties, and I'll show that on the next slide with the counties that we've been finding it in. We only found it in 3 counties in 2023, which we think was largely due to drought. And then 2024 was kind of our standout year. We found it in 46 counties and then in 23 counties in 2025. This is a picture from a South Dakota field. And you can kind of see right here our different tar spot lesions. And one of the kind of interesting things about tar spot here and in a couple of other states is that we actually don't see the characteristic fisheye lesion that a lot of states report. So the fisheye lesion is the brown necrosis region that appears around the black lesions. And I have a couple of examples of that in the next couple of slides. But that's been something interesting that we've been kind of watching develop over the last couple of years here in South Dakota is that we're not really seeing a lot of fisheye lesion, and it's kind of an unknown of why. So this is going to be one of our spread maps. The blue line here, that's where in our southeast corner that kind of touches Minnesota, Iowa, and Nebraska. That's where we found tar spot in very limited quantities in 2022. And then the purple counties here, we found that in 2023. And then these orange counties that you're seeing were our 2024 finds. So that was that year that we found 46 counties that were positive for tar spot. Tar spot is generally more severe in this state, and as we go north and east, it becomes much, much less. The next slide. In 2025, we did start finding tar spot again in these typical southeast counties right here. And I kind of put some dates on here to kind of give a context of time. We found the first 2 reports of tar spot in July on July 25th. And that's a lot later than in other states, but it was much earlier for us in South Dakota, where our previous first report in 2024 was September 1st. So we were about 35 days before we had ever found it in the last couple of years. And so also we had our first report of tar spot coming from residue that was previously infected. And that was something that we had kind of questioned, would the tar spot be able to survive the cold here in South Dakota? Would it, be able, or was it just going to continue to be a windblown disease? And so for us, we definitely were able to start seeing some effects of infected residue being left in fields that were corn on corn. So again, these first counties were found in July. And then tar spot kind of worked its way here, and you can kind of see the other counties that border us. So we were kind of matching them. And then it started going west. October 16th is the last report of tar spot that we received, and that was from Sully County. And it was it was not very severe. It was just a few lesions in the field. So for us, and that kind of follows our typical pattern, it starts here, moves up to the north, and then kind of to the west. And whenever we look at weather data, it very much explains how and why that's happening, because it's usually the combination of heat and humidity that starts here, and then we kind of start seeing it around the rest of the state. And if you've not seen tar spot before, here's some different examples of it. This is more typical of what we're seeing in South Dakota, where we have 1 or 2 big lesions and then a bunch of small lesions. This is actually an example from, I believe, Missouri, that I had taken whenever we went on a scouting field trip. And then this here, if as you may have heard me talk a few slides ago, talking about the fisheye lesions. So you can kind of hopefully see these here that have the brown necrosis around the lesions. We are not seeing that in South Dakota yet. And so that's one of our kind of ongoing research projects of why we're not seeing it. What's happening there? And I don't know if we'll ever find the answer, but it's definitely something that we're kind of curious about and why it's looking a little bit different here than what it is in other areas. So, as we think about management considerations, here in South Dakota specifically, tar spot as a whole is still appearing later in the season, generally after the major reproductive windows where fungicide would be useful for control. However, we do note that it is starting to occur kind of in those far southeast counties a little earlier every year. As again, it was found in July of 2025. So we are starting to recommend that our producers consider, looking for hybrids that might be showing some signs of resistance or tolerance, and be considering putting out an extra fungicide application for control of tar spot. For us, it's still the amount that we may have and how severe the infection may be is still very hard to predict, because it does appear that most of our spores and infection load is still windblown. And so we're still kind of dependent upon it getting started in other states and blowing up to South Dakota. And we also, these are the counties here that we have been kind of targeting for people to start looking at. And that's mostly because they border other states and they've had, 4 years now of pretty heavy infections. And yeah, so that's kind of our tar spot story. We are going to start trying to do more specific field screenings of varieties and possible fungicide control over the next couple of years. But we are going to be doing all that with natural infections. So we are hopeful that we'll start getting some answers and maybe also be able to develop a understanding of what tar spot's going to do in South Dakota. One of the things that we've heavily used here because we're not fully understanding when tar spot may show up, is we've been using the crop risk tool from Crop Protection Network. That has been very insightful to try to help teach and understand about spray timings, when we should be worried about tar spot, and all of that. And so these were just some of the towns that we just kind of randomly selected in the eastern side of the state this year. And you can kind of see this one here, tar spot was mostly low to moderate, the risk of it in the susceptible time frame. These are similar results. This town here was kind of in the southeast corner of the state. And so while we did see a lot of tar spot in the southeast corner of the state, it was later in the year. So this really kind of lined up nicely on ground truthing that the risk period for tar spot was really low. And we actually didn't see a lot of tar spot until later in the year. And while some plants didn't look super great, we don't believe that we had any yield losses either from it. And then this was kind of one of our more interesting pictures that we've gotten. And if you've seen these lesions before, those are southern rust. And so this has been a very common occurrence in fields, and I know for other states too. But we'll find these really heavily infected tar spot leaves, and then also find southern rust. And it's just kind of been one of those interesting things to see that the two diseases are co-infecting with each other a lot of times on the same plants and the same leaves. So I just love showing this picture. So now moving into another corn disease that we've been working with, or possible corn disease, we're not really sure. So one of the things we have been working on is understanding BLS a little bit more. How is it spreading across the Great Plains? When is it starting? Where is it at? And so over the last 3 years now, because we haven't finished up our 2025 data, but we did collect in 2025 as well. But over the last 3 years, we've been collecting corn leaves that were symptomatic for BLS from mostly South Dakota and Nebraska. We have gotten a few from Kansas and a few from Minnesota. And so we've been collecting those samples so that we could see what the BLS looks like, understand it, and get to maybe have a more fundamental understanding of how this disease is spreading and why we're seeing it just kind of randomly all over the place. So these are our sample areas over 2023 and 2024. And yeah, so this is kind of the results of us identifying the bacteria. And you can kind of see here, Xanthomonas, which is the primary cause of BLS, we rarely ever found it. We only had one positive sample in 2024. We do have a couple that we think may have been positive for Xanthomonas, but they were kind of overgrown. And so by some of these other bacteria. But as a whole, a lot of what we're calling BLS is not actually the BLS documented to be caused by Xanthomonas. What we are actually finding the most of is going to be Pantoia. You may have heard of Pantoia before because it is a cause of Stewart's wilt. And so this is a different, it's a cousin to that disease. And so if you've also may have heard of Pantoia in the context of causing another disease in Texas. Over the last couple of years, Texas has documented what they've termed late season decline, caused by probably Pantoia ananatus, which is this one right here. And they've been finding that it's been causing a lot of issues. It's been causing stunting, yield loss, lodging, all sorts of different things. So with that report, we had, and our results, we started wondering why we weren't seeing the same symptoms. So we've been collecting all of these bacterial samples over the last few years to try to really dive into, why we're seeing the same bacteria that are responsible for this fairly severe disease down south, and it's not doing a lot up in Nebraska, South Dakota area. And we're still working on that. We do have a few theories. We think it's mostly based on timing, but I can kind of show you some of our information here. So these look really big and scary, but they're actually not too terrible to kind of follow. What I mostly wanted to show is we've marked all of our samples with the state that it came from. And what we're seeing, especially like this group right here, so these are all genetically related to each other, is, we have samples that are all in theory the same that have come from South Dakota, Kansas, Nebraska. And we'll find that a lot. We have South Dakota, Minnesota. So these bacteria that we're finding are fairly widespread. And they appear to be, kind of the same genetically in a bunch of different states. So that's one of the things that we've been kind of working through, is kind of looking at how they're related to each other and why we might be kind of seeing them all over the Great Plains area. And so these are another set here. And again, just really showing that we're seeing what is in theory the same bacteria across a bunch of different states, which is, very interesting to kind of think about. And then this is the Pantoia ananatus tree, which is the one that was reported for late season decline in Texas. And we can kind of see that we have similar bacteria, but we're not getting an exact match to the Pantoia ananatus that was reported in Texas. We're not sure if that's just that we need to dig a little bit further into the genetics, or if it's that we may have more Pantoia agglomerans. But all of this goes to be summed up as we are seeing the same bacteria, and but it's not causing the same symptoms. We've had a lot of questions here in South Dakota. We have companies that are doing soil testing and plant residue testing and finding Pantoia ananatus and Pantoia agglomerans. And we have a lot of people that have been concerned, producers that have been concerned over the last few years about why that's happening and what does that mean. And so this is kind of the start of that research. Unfortunately, we don't have a ton of answers, but we are doing a lot of work on it. It's kind of our main focus right now, because we want to understand, is this disease as bad as it appears to be in Texas? Or is it truly that because our environment and our farming systems are pretty dramatically different between the panhandle of Texas and South Dakota, Nebraska, if we may not see the same bacteria or the same disease symptoms just strictly based off of environment and timing. So this was all of the Pantoia species. So all of those trees that I just showed you of all of the bacteria that's related to each other. This is kind of where we found them. And again, this is just kind of a way to show that it's widespread. We're seeing it all over the place. And we are finding other bacteria, which you saw in that previous tree. And those we just think are, kind of saprophytes and bacteria that just kind of lives on plants. But we are definitely keeping an eye on this, and we'll hopefully have a few more answers in the next year as we really get this research finished up. So now I'm going to swap from corn to soybeans. We have been also, like many other states, tracking soybean cyst nematode and what it's doing in South Dakota. When I took this position, we knew that we had soybean cyst nematode in about 30 counties all on the eastern side of the state. We knew that we had about 25 or so % of the populations were becoming resistant to PI8788. So, they're feeding on, the nematodes are able to really feed on those plants and reproduce and cause yield loss. And so I just kind of want to set the stage for that, for what I'm going to be going through. So this map here, I don't know if you can kind of count those really quickly, but that's way more than 30 counties. We are now to 49 counties are positive for SCN. Over the last 2 years, we've confirmed the counties that are kind of in this pink color here. So one of our biggest finds is that SCN is really getting west of the Missouri River. One of the things that we always refer to is East River and West River in South Dakota. And so we're now finding several counties that are positive for SCN West River. And this was not something that we expected. However, these counties are newer to soybeans, probably in the last 20 or so years. So it wasn't completely unexpected, but we didn't, we weren't really thinking that we were going to just find all of these SCN. So basically the way that this map reads is the first report of SCN was here in Union County in 1995. Then the green counties came next, blue, purple, pink. So you can kind of see how it's moving across the state kind of in waves. So yeah, that was something we just finished up this research in the last couple of months, and the student that was doing this is graduating. So I have fresh data from her to be showing you. We have 2 major HG types that we found in South Dakota. And if you've not heard of HG typing, that's heteroglycines typing. So it's looking to see how those nematodes and in what quantities the nematodes are feeding on our resistance genes, such as Peking and PI8788. So we are finding that we have a lot of HG type 1, 2, and HG type 2. So again, that means that the nematodes are reproducing at some level on those resistant plants. So HG type 1-2 is by far the most common HG type in South Dakota, which is a little frightening to think about because type 1 is peaking resistance and type 2 is P8788. So, what this means in a larger context is that we are losing genetic resistance that is able to manage the nematodes. So, rotation is going to become infinitely more important here, rotation of both crops and of resistance genes, because we are documenting a lot of type 1.2 and type 2 resistance here. And this is just kind of another way to like look at this data. So this is peaking, so type 1. We can see that we have a lot of populations that are just kind of at that break-even point of being able to feed and reproduce on plants and cause yield loss. So that's what this line is here. So vast majority of our populations are kind of in that lower level of resistance. However, with type 2, we can see that we don't have as many populations at the break even, and we have a lot that are kind of creeping up into here. And if you've worked with HG typing, you know that 60 kind of means that the population is totally resistant to being managed by resistance genes. So you can see that roughly half of our populations that we did and worked with in this study are totally susceptible or totally immune to resistance genes. So they are not affected by them. They're not managed by them. We've kind of lost our ability to use PI8788 in a lot of context with a lot of those populations. And then this is kind of, again, showing the same thing I was talking about. So here's that 60 mark, and the orange bars that you're seeing are going to be populations that have gone over the FI index, so the female index of 60, and are basically not being controlled with resistance genes anymore. And so all of these are counties here. We incorporated, I think, about 40 counties into this study. So we tried to get just as many populations of SCN as we could to try to do a good, thorough survey. And then the blue bars that you see here are the peaking resistance. So kind of a little bit of doom and gloom on our side of what SCN is looking like and what our HG types are looking like here. And with that, we thought that we should take some time to maybe develop a little bit faster of a way for detection of SCN. So you may have heard of PCR before or field-based testing or something like that. So our most typical way to identify SCN is going to be through morphological identification through a microscope, which is what you're seeing here. And there's still value for that, and it's still very necessary because that's the only way that we can count the number of eggs and cyst and know what your numbers are when we do a soil test. However, sometimes we need a little bit of a faster, quicker yes or no. And so we have been developing some diagnostic tools to try to get that yes or no. So we have developed a couple of assays. We've developed a real-time assay, so we get an answer of if it's a soybean cyst nematode in 1 to 2 hours. We also developed an RPA assay, and RPA is what is called a on-site or field-based diagnostic tool. And so that's been kind of the big buzz in the plant path world, especially in the plant path diagnostics world, is trying to develop these tests that we can do faster. They do lack sensitivity, but you do get a quick yes or no. So we've developed both of these over the last couple of years. And so this is for our qPCR assay, and this is basically just showing that it works really well. It's very sensitive. We can detect pretty low amounts of DNA, which is really exciting. So we can, hopefully be able to test soils and at least get a yes, no that we can give back to those producers while we're doing their counting. So these are just some of our different tests that we did. And just basically showing that it works really well and we can detect small amounts of DNA. And then also, we were able to make sure that not only is our test sensitive, but it's also exclusive to only SCN, which is another really important thing that you want to check whenever you're working on these, because we want to make sure that we're not giving a producer or a crop consultant a false positive. So we did some work to make sure that we weren't amplifying any of these other common nematode species, and we did not have any issues with that. So we were really excited about that aspect. And so, yeah, that's kind of our qPCR. We're very excited. We'll hopefully have that out for use next year. And then moving into the RPA assay, so that field-based assay, we also developed a protocol to get this going, and it does work really well. And so, kind of what we would love to do with this is be able to take this when we go out to meet with a producer and test some of his soil and quickly say, yep, you have nematodes. You need to send that soil in for egg counting to the diagnostic clinic. Basically, to try to give a little bit more reassurance that they're not wasting their efforts on sampling soil and sending it to the clinic for egg counts. So we do lose some sensitivity with the RPA tool. So we were able to identify 1 times 10 to the fourth, so a very small amount of DNA with the qPCR that we can't identify with the RPA. However, again, this one we can do in the field. qPCR has to be done in the lab. So there's kind of, risk and benefits with both. And we actually just finished this up about 3 weeks ago. And we were able to verify that both the qPCR and the RPA can detect nematode DNA down to 1 cyst in soil. So we're really excited to be able to get these out into the field and show producers how we're using them and how they can kind of help supplement what sampling methods they may be doing, or at least give a, yep, you have an SCN problem. Let's get that soil, sampled and counted for eggs in the lab. And with that, that is kind of a big update on what we have going on here. We've got some other work going in wheat, but I felt like corn and soybeans were enough to talk about with all of their different diseases. So here's my contact info if anyone needs it or would like to reach out.